Okay, next up we have Aashiq Kachroo from the University of Texas at Austin, and he'll be talking to us about deciphering common principles governing gene replaceability in yeast.
Thank you. First of all, many thanks for inviting us to talk about our work. I'm going to bring in 3 more model organisms to this forum: bacteria, plants, and also humans. So and you'll learn by swapping these genes in yeast, how we learn about gene evolution and their functions. Okay. So the fundamentals of this whole project stem from the fact that we share genes with nearly every organism on Earth, and that includes distantly related organisms like fungi, over a billion years of evolution that separates us, including bacteria. So we have, our ancestry is deep-rooted in evolution. But what we wanted to test was how many of these genes, what we call as shared genes, are truly functioning the same way as in humans or in other species. So we published this work recently, but I'll give you a glimpse of what we showed there. So we ask a question: if you take a gene which in yeast, knock it out or delete it, so you get a lethal phenotype. We look for only lethal phenotypes, so the cells don't grow. But if you take an orthologous gene from humans, put it on a plasmid, express it in yeast, and the cell lives. So if the cell lives, the gene functions the same way as the yeast gene is doing. It doesn't, so therefore it doesn't. So this was a simple test we used. What we did was, it was a large-scale study. We looked at over 400 genes which have clear one-to-one single orthologs in either species. And we found nearly 50% of the genes from humans can functionally replace their counterparts in yeast. All right? So from there, we went on to learn about principles of replaceability. What does it mean by functional replaceability? So we looked at several properties. If you want to go delve into it in detail, you can actually look at a paper. But I want to give you a glimpse of one major thing that we found. What we found was this replaceability, or replaceable genes or non-replaceable genes, were not just scattered around in different networks and different pathways. They actually had a system-by-system bias, wherein we had a pathway like proteasome or cholesterol, where we had large replaceability. Yellow is replaceable, blue is not replaceable. And on the contrary, we had systems like chaperone complex or origin recognition complex where nothing was replaceable. So there was a strong bias, system-by-system bias. We also found the sequence similarity and identity was not necessarily the best property, although there was a signature which explained some aspect of sequence similarity contributing to replaceability. All right, from there on, we asked, so what we did was we looked at 1 billion years of evolutionary divergence of separate evolution. We wanted to know whether we have learned general principles of replaceability. What if we go now to bacteria, which are roughly over 2 billion years of divergence? So bacteria still share genes with yeast, over 400 of them. And we asked the same question. There are 460 orthologs, distinct orthologs in bacteria. Of those, 60 are essential in yeast, so we can actually do the same test again. And we looked at only those which had single distinct orthologs and did the test. We PCR amplified them. It made it easier because bacteria don't have introns, so we just clone the gene. We put them in yeast vectors. We express them constitutively in the yeast and did this assay in 2 different strain backgrounds. And here is the crux. We found over half of them functionally replaceable from bacteria in yeast, a prokaryote into an eukaryote. All right? And what we want to get from here is the red is the replaceable gene, the bacterial gene, and the dashed line is the wild-type yeast strain. So they are very comparable in a YPD-rich medium. They have very little growth defects. So this is amazing. And further, we wanted to develop into another aspect which is different between prokaryotes and eukaryotes. Eukaryotes have subcellular compartments, and one aspect of that was very evident. We had nearly 11 genes which are supposed to work in mitochondria. And what we did was we thought maybe that's important. They're supposed to be mitochondrial localized. So what we did, we engineered these E. coli strains, genes, with the mitochondrial target sequence and said, now will they function? Turns out of the 11 that we tested that way, 4 of them now started functioning when we put them in the right compartment. 2 of them in the same pathway, which is branch chain amino acid pathway, ILV-C and ILV-D. And one of them is a chaperone, which works from humans, also works from E. coli. Highly conserved protein. It doesn't really matter whether you take the gene from E. coli or you take the gene from humans. We also had a ribosomal protein working now when we put them in the correct compartment. We showed that when we do this engineering, where we put this mitochondrial target sequence, they indeed go to mitochondria. So this is an experiment for that. This is a protein tagged now with the EGFP, and we look for mitochondria by mitotracker red, and they co-localize and show that this is indeed going to mitochondria. Now, with these sets of genes, that nearly half of them which replaced from E. coli, we wanted to learn principles of evolution again. So we looked at different properties of these genes, sequence, codons, network, many properties. What we found was, like in our earlier paper, that network features came up as predictors again. So in a nutshell, what I'm showing you is the predictive power on the y-axis, which is in the form of AUC, or anything significantly more than 0.5 is predictive. So we find one thing which was different. Less optimized codons. So genes that replace had less optimized codons for E. coli expression, E. coli. We found less connections in protein-protein interaction networks. So the protein will function if it had fewer connections or interactors as compared to many interactors. You also find that more sequence similarity. So sequence does have a signal. But again, if you look at the graph and look at the x-axis, which is basically a binning in these proteins on the basis of sequence identity, you find that at 25%, I will have significant number, but the functional replaceability is at 0.4, so it doesn't tell whether the gene can replace or not. But as you diverge or you go more similar, you actually lose functional replaceability. And at 10 to 20%, we had nearly everything complementing. So again, sequence doesn't necessarily tell you whether the gene can function or not. I want to go to our champion replacer now, a champion pathway. This is heme pathway. Heme pathway is highly conserved. So only few parasites have lost it. And so in yeast, this pathway actually goes in two compartments. Starts off with mitochondria and ends at mitochondria. All right? And here's the data for humans. So dashed lines indicate paralogs. Nearly every gene, not nearly, every gene in this pathway is able to function in the context of yeast. And in the E. coli. In E. coli, we had two genes which are indicated as diamonds, which are not clear orthologs. We don't have any information that they are orthologs or not. So we call them non-orthologous, and even they replaced. So E. coli genes also replace, irrespective of the localization, right? And we then went into plants. And it turns out plants, this pathway is also conserved and makes chlorophyll. All right, and it's entirely chloroplast localized. And plants also work. Plants have many duplicated genes in this pathway. Majority of them worked, except for one reaction which is at SCHAM12. But plants replace, E. coli replaces, humans replace. So I think we have now found a universal replaceable pathway, which is heme pathway. Integrating, we also used at one point other species like soybean. This is an important pathway because people target it for herbicides as well in plants. Also, we wanted to know whether plant genes, which are supposed to be chloroplast localized, do they localize to mitochondria? Turns out, using the same method I showed earlier, that they indeed localize to mitochondria. So there's a significant crosstalk between the target sequence of the chloroplast and mitochondria that sends them to mitochondria in yeast. Okay, so this pathway has a very interesting history. We started off from the ancestral pathway. We acquired and became eukaryotic-like, but acquired this at two events where due to endosymbiotic events, you have mitochondrial acquisition, plastid acquisition, chloroplast acquisition, and then plants became bacterial-like, eukaryotes and fungi. Humans and fungi are eukaryotic-like, but what I showed you that it doesn't really matter. You can swap genes from bacteria into yeast, from humans into yeast, from plants into yeast, and we have results show now that you can actually take eukaryotic genes, put them in bacteria, and they also work. So this is like a universally swappable pathway. And in a nutshell, what we have is roughly over 60% of the genes from E. coli working in the context of the eukaryote yeast, and this is remarkable. If you want to go further, develop further into this pathway, you can go to the posters, 2 posters we have, John Laurent, where we show that we have now replaced all the essential genes that can be swapped in yeast. That amounts to another 400 new genes. These genes have very special in a way that they have duplicated, have paralogs, and we learn, while we do and make these pathways humanizable in yeast, we also learn about the evolution of duplicated genes and how they function. And you will also learn that we have now humanized, on course to humanizing the entire proteasome core. We have now strains using CRISPR which are expressing 5 humanized proteasome subunits, alpha proteasome subunits. And the similar vein, we are building pathways like sterol and heme pathway from bacteria and plants as well. And if you want to get a little more curious, we have an equalized version of yeast, which is pink, and we think we have probably a porphyria in yeast, which is a disease which causes porphyrins to secrete out porphyria. All right, so summarize, humanization, plantization, and bacterialization of yeast was successful, and this we have now tested with 1,000 orthologs, and nearly half of the time it actually works. Remarkably, prokaryotic genes also work in eukaryotes, and like in yeast, and we show that in some cases you really don't need a mitochondrial localization signal, but some cases you do, and this successful replacement of is again system-by-system basis. We find pathways which are entirely replaceable, pathways which are not, and we found the heme pathways a universally swappable pathway. And that finishes my talk. I want to thank my advisor, Edward Marquardt, his amazing lab, and I have a complete freedom to do many, many different experiments. John Lawrence, the current, finished his PhD in looking for postdoc, an excellent student. He was on this project. Azad Akhmatov is another guy who's also having a poster and wonderful graduate student, and many, many Markosians, diverse lab. Klaus Wilke, we collaborate with to learn about in silico evolution of these genes, many undergrads, funding from CIPRIT, NIH, NSF, Welsh, and thank you for listening to me.
In this universally replaceable pathway, if you have phenotypes associated with certain mutations in an organism, do you also have phenotypes with those mutations in the replaced pathways?
Oh, this is very interesting. So, so you're saying if the gene has a mutation and leads to, for example, disease in humans, would that mutation have some phenotype similar in yeast? So this is a very interesting question. It goes to another thing which we are studying in the lab, which is called orthologous phenotypes. So if you look at plants, they make chlorophyll. So the mutation will have impact on the chlorophyll synthesis. In humans, this makes hemoglobin. So mutation will have impact on blood and different phenotype, totally. So you can have orthologous phenotypes, and yeast, it will have growth phenotypes, typically. Okay. Or it might actually start secreting out these porphyrins and have similar to human phenotypes. So you can get different types of phenotypes. You can read them out differently in yeast. We look at mostly growth phenotypes, but we can, look for different other phenotypes. So because the pathways eventually lead to different outputs, you might have different phenotypes in different organisms.
Okay, because it would be very interesting to heavily mutate, for example, this universally replaceable pathway and see all of the consequences in all of the organisms.
Yes, absolutely.
Okay.
So I was wondering how many of the genes that you can't replace are actually translated into proteins? Have you tagged the human versions or the E. coli versions to see if they're actually expressed? Because you noted the codon biases.
Okay, so if you follow our first paper that we have published already, we did a small scale experiment where we looked at Western expression of these genes with HA tagged and showed that nearly all of them that we just, looked for expression where indeed expressing, it doesn't matter whether they are complementing or not. So yeah, we have done that earlier.
I was interested in the complementation of the ribosomal protein deletion. How many ribosomal proteins have you been able to complement?
This data is available in SGD. So if I recall, I don't think many ribosomal proteins were replaceable, but there were some which were replaceable.
So 2 questions really. So one, replaceability and how does that correlate with cell cycle regulation of transcription? And in the case, and a kind of a follow-up on that, in cases where you have a catalytic subunit and then a regulatory subunits, do you see divergence in replaceability, for instance, like in cyclin CDKs or other complexes like that?
Okay. Regarding the first question, the cell cycle, we found that many cell cycle proteins were not replaceable, at least. It was a small subset, but they were not replaceable. So we had a couple of reasons, but so one of the reasons is that there is a cell cycle kinase and a CDK, which regulates the kinase. It turns out that you need both of them, so they have become specific for a certain organism. So yeast has probably a different version of these, and they are very strongly regulated. So that might be the reason why we couldn't get many cell cycle proteins replaceable. But some of them also are not essential, so they were not a part of our set. The second question you asked was whether that is the reason, the regulation is different. Is that what the question is?
Or regulatory, is there a difference between like the catalytic subunits and regulatory subunits in cell cycle complexes like cyclin CDK?
Well, I don't know the answer to that exactly, but yeah, I have not followed that up. So thank you very much.